Thanks for allowing me to talk about CryoEM during this symposium. I'm not sure if you're all here because you're really interested in CryoEM or because you're still sticking around for the reception after, but I will give you a very quick introduction into what CryoEM is and how we use it, and then Jason will talk a bit more about all the data we generate and how we deal with it. So my name is Mark. I joined Rockefeller in 2015. I used to work for the Electron microscope manufacturer. I came and installed the microscope here, and then they invited me to stay.
Did they chain you to the microscope?
Yeah, I came with the microscope. FBI at the time did not expect that to happen. And Johanna is my assistant. She's a research support specialist, and together we operate the microscopes and help everyone collect data, and we're hiring one more. So if you know a talented cryo-EM scientist, let me know. The instruments we use are electron microscopes. What they do is they take an electron particle beam, they accelerate them at different voltages. That gives you a different wavelength, and you have incredible resolution. Every time there's a light microscopist in the audience, they're like, yeah, well, you get good resolution, but you're wrecking your sample. This is partially true, and we have kind of ways around it. But with the Arctica, we get 1.4 angstrom. That's really small. And with the Cryos, we get 1 angstrom resolution, and we have a third microscope going in now, which is a fully loaded Cryo, so it has all the bells and whistles and options you could possibly need for life sciences. I just had a daughter. She's one year old now, and I'll typically see Jason in the hallway. He's like, "Hey, how's your baby?" And I say, "Oh, Maria's doing great." He's like, "Yeah, I know your daughter is great, but how's your baby?" I was like, "Oh, the microscope. Yes, the new microscope is spectacular. Here it is. This is a G3 Cryos. You guys will see it when we go on the tour." So what do we do? What we do is we have a complicated sample preparation technique called vitrification. It is complicated, but if you see it done, it's actually a miracle that it even works. It's kind of like an ad hoc way of getting your protein embedded in ice. But after you get your protein embedded in vitrified ice, you expose it, and you will get this micrograph, typical micrograph over here. Is there a laser pointer? Yes. You get a micrograph. When the electron beam irradiates the ice, there is beam-induced motion. And we have these high frame rate direct electron detectors, which can very quickly take many frames in what we call a movie, and you can motion correct and align for the movement. So if you have movement, you get a blurry image, that's bad. You motion correct, then you have a good image, or a motion corrected image, with all these particles embedded in vitrified ice. And so you can imagine that if I have many, many duplicates of the same exact protein, and they're all spatially oriented in different ways, and I can see them from all different sides, I can then tell the computer, hey, here's a particle, here's a particle, here's a particle. I select the particles. Some of that's automated, some of it isn't. And I can then finally reconstitute a 3D structure of my protein. So if you are in the field of structural biology, you know that this is the most important thing that has ever happened in science in probably the past few years. And if you're not in structural biology, now you know that this is the most important thing that has happened in science in the past few years. So we generate terabytes of data. And then we have to keep the raw data, because sometimes you will align your movie, and then you get let's say a 5 angstrom reconstruction, and then somebody's like, hey, wait, if you align the movie this way, you can get a 4 angstrom from the same exact data. And so we really need to keep all this raw data, because people are coming back and realigning and reprocessing and finding that they can get much better data. So in terms of what we do for management, Jason will talk a bit more about it. I'm mildly involved in it because it lands on my workstations, and I've screwed things up, and Jason has saved me, and so he'll tell you more about how we handle our data. But that's what CryoEM is, and thanks a lot.
Okay, great, thanks. Yeah. I think Mark gave a good overview of the initial steps of what happens and what the end goal is. I think one of the things I wanted to convey here is that I think there are really good opportunities for data management. We asked who has a data management plan, and it seemed like things were very ad hoc. There are really great opportunities for data management when a core facility or a resource center or whatever you want to call it in your institution that operates these high data rate instruments gets together with the IT group and makes a solution. And that's one of the things that I think we've been able to do here in the CryoEM space. So this talk is also just a very quick preview to the tours that we'll give of the data center that many of you have looked at through the window and the CryoEM facility. Essentially, the processing that happens in the CryoEM data after it's collected lands in that infrastructure over there. The vital stats are there. I think this is probably the smallest system that's been presented today. We're a small institution, but it is what we have. This to me is the money slide. In that I think there's a strong argument that raw data can be kept in perpetuity. And in perpetuity is in quotes. I'm not sure about 50 years, but 5 or 7 are numbers that are bandied about for regulatory reasons and so on, but also for scientific reasons, this idea of going back and getting a better structure, also the ability to go back and mine data, right? If we're going to write or develop neural networks that are going to do particle picking, it's great to have a lot of raw data that you can train on. So I think these raw data sets do have value. One of the nice things about raw data that comes off an instrument, it is kind of by definition immutable. So that makes data handling a lot easier. You can leverage that in terms of economics and other ways. So what our groups, Mark's and I's groups, have been able to do is sort of cross out that line that you see in a lot of resource centers. That's data will be kept for 2 months and then we're going to delete it and it's your problem. And people come with USB drives or they have shelves full of Drobo boxes or something like that in their lab. And one of the things that makes this possible is my infinite series there. Basically, the math is saying if A is the cost of storing a terabyte of data today, right? One of the things that if you just analyze the pricing in the industry over many, many years, storage price per terabyte drops about 20% per year. Sometimes a little more, sometimes a little less, but that's a pretty broad trend, right? That's just capacity. We're not talking about IOPS here, we're talking just capacity. But when we're talking about archive, that's really the operative number. So if you know that the price of storing that data is going to go down by 20% per year, and you recognize that once you're done with that data in a high performance HPC environment or something like that, and you can move it to a cheap and deep system, you can often save, our model says it's about 1/4 of the price of high performance data. That equation gives you the cost of keeping that data forever, right? That sum goes out to infinity. But since the price is going down by 20% per year, that actually converges. So it's maybe counterintuitive that if you keep data forever, it's not an infinite cost, at least for the hardware, right? So yes, having 20-year-old data on spinning Rust, that what was big 20 years ago or 5 years ago is not so big anymore. And in many cases, it is not, I would argue, is not worth the human effort to go through it and to decide what to delete. Right? You can make an economic argument that when you factor in people's time, it may be better to keep. So I think that's can be a compelling argument to make that case. That said, leaving lots of files on disk, a 20 petabyte POSIX file system with files randomly strewn about it is not something that's great to manage, and some sort of curation is important. And what we've done with the CryoEM is basically capture every bit of data that comes off the microscope and just put it in a very simple structure. It's got a year, and then within the year it's got labs, and then within the lab it's just a unique identifier for that run. One of the things we see here is, or a bit of opinionated advice is if you go for something like this where you're going to just curate data that comes off an instrument, is don't get too clever with the, say, directory structure or whatever. There's a tendency to build in metadata into the directory structure. It's this protein or it's that sample or whatnot. That has always bitten me in the bottom later because you find out, well, it wasn't actually that sample, or it was something else, or the metadata that I care about today is different than the metadata that I care about in a year from now. So just a unique identifier that uniquely identifies the sample, that's immutable, and then keep a database somewhere else where you key off all the other information. if you work with a core facility, it's a great place to capture some of that metadata because you know it at that time. And in a core facility, you can work systematically. If it's an instrument in a lab, it's going to be very difficult to get the lab to enter that metadata. But in a more structured environment, that's going to be possible. So those are things that have worked for us in this model. Just some results. This is what our archive looks like in terms of size over the last year and a half. So each little block is a particular run. You can see that runs have actually gotten bigger. We may actually be doing a little bit less of them per month, but they're bigger. And you can see since the beginning of this year, we're generating about twice as much data as we were in the past. Some of that has to do with the fact that we're introducing collecting data with beam tilt on our microscopes, so you actually can collect more data. It's like getting a free cryos. And some other optimizations that have been made. So that's just sort of what the growth of our archive looked like. And I might infer from this that we do maintenance in February, or it's just too cold and people don't come to work. But some other data that we've collected. We talked about just how to slinging data around campus, or what the data rates coming off these instruments are. We actually instrument our machines in quite a detailed manner. Here I'm just looking at some bit rates off of network card. We actually collect this data every 10 seconds and keep it forever at full resolution. So it's really helpful for troubleshooting and things like that. Here you're looking at a few days of data. But what we see here is that in a CryoEM run, there may be multiple hours or a day-ish or so where we maintain 20, 30 gigabytes per hour. That's actually not what I would call a super fast data rate. 10 gig network, that's cool, that's fine. But if that keeps coming at you forever and ever and ever, that's something you need to know. But here we see it's kind of bursty. That's on our Arctica. In the early days, the Arctica was considered the screening microscope, right? So I think we talked about, I think Stefan mentioned, maybe get a less expensive instrument that you use for screening, so your high quality instrument can be, you can really not waste time on. So Arctica was often used for screening, although lately we've actually been using the Arctica for data collection as well. So that's another reason why some of the Arctica runs have gotten bigger. Our Cryos with beam tilt is pushing out 60 gigs an hour, and we're running that for longer periods of time. So this is the kind of data that you need to do your capacity planning and so on. This is just kind of a schematic of what the data flows look like. We do have these image acquisition PCs locally. I'm a big fan of that. So when I saw the suggestion that maybe you push data from the instrument, across the city into a remote facility, I get a little skittish about that. I've just found, at least where I've worked, institutional networks have their flaws, and depending on that, when you have precious samples that you can't run again, can be a risk. So I kind of like the idea of local data collection. One of the things we found here was, so we were essentially doing that, and originally we were pushing data from these local workstations to the centralized archive in near real time with periodic rsyncs and whatnot, and originally found that actually just broke the campus network. That would happen, and email would stop working, and Anthony would yell at me, and so on. So one of the things we did was, I guess the advantage of having a small campus is we actually just did dark fiber from the back-end HPC network right to those workstations. So in some sense, you could say, even though physically that may be far away, it is sort of a direct connection, in that we're not going through all our routers and firewalls and so on. That's just back-ending that right in. And then that's sitting on its local compute, so you can analyze the data in situ. I just wanted to end with one of the things that taught us, though, is that we can't do that forever, right? We can go to a CryoEM facility and say, okay, we spent X million on a microscope, we can afford to pull a piece of fiber along with it, but there are so many high data rate instruments that are being delivered to labs, and we can't pull fiber to every single lab. So what this actually caused us to do was embark on a campus-wide complete revamp of the network. And it's not just an upgrade of the core from the Cisco Catalyst X to the Cisco something Y, but it's a real rethinking of the architecture to support science. And part of that is, this is a proposal, we're not sure we're going to do it this way. But first, we've got lots of red lines with 40 gig links running around where we're currently at 10 or sometimes even 1. It's just the way things evolve. But really, one of the key elements here is that we're trying to make the campus a bit more of a mesh so that traffic that needs to go from one building to another doesn't necessarily have to all bottleneck in a single core. So that's, I think, a really important strategy as we grow and anticipate that this is going to be a problem we're going to have to solve multiple times. So if we can mesh it, we can always create a new path when needed if we have the right kinds of monitoring and so on. So what started out as a small data movement problem just turned into a 2-year major capital project. But I think it's the right thing to do. And again, this is something that getting an institutional network group to work with a science group is not always the way institutions work. But it's something that we were able to achieve here. And I think that has some benefit. And I would encourage making that happen whenever possible.